Good morning. Thank you for coming. My name is Jonathan Garen. I'm the Chief Corporate Development Officer at Ensoma, and I will walk you through an overview of our company and our programs, platform, and technology. So just by way of introduction to the company, if you're not familiar with it, we are an in vivo hemopoietic stem cell engineering platform with several programs in development and one on the cusp of getting into the clinic. So starting with a little bit of who we are, this is our beauty pageant. We are the leadership team on the left and our founders in the middle, Hans-Peter Kiem and Andre Lieber at the University of Washington, and the Fred Hutch that developed some of the key technology that went into forming the company. The investors as well on the right-hand side. We have venture investors, we have essentially crossover investors, and we do have corporate and strategic investors as well. So the overview of what we do, we have a proprietary delivery particle that was created by our founders. That is a virus-like particle, and it's really the heart of what we do because this enables us to very safely and selectively target the long-term hemopoietic stem cells in vivo and deliver very high-capacity payloads to the nucleus, so gene insertion. And that's something that no other company can do, and there are products that I will describe that no other company can create and no other technology can do. So this is very important just to understand fundamentally what drives the company. It enables us to create any combination of modified hemopoietic stem cell derived cells in ways that we want, and I'll show you what we mean by that specifically. And we are a company about to enter the clinic with an IND submission in the first half and clinical readouts actually that will occur in the second half of this year because our clinical trial has quick readouts. So just as background, as a reminder, the hemopoietic stem cells are the stem cells for all the white and red blood cells in the hemopoietic system. They are shown on the left of the slide, and they are a self-renewing source that can be differentiated into many different lineage cells. I'm sure many people are familiar with, B cells, T cells, monocytes, platelets, a wide variety of different things. So by engineering the hemopoietic stem cells, that engineering will affect all of the progenitor, all of these different lineage cells, but we can selectively turn on just the ones we want in any combination of these. And again, this is extremely important because we're the only company that can do that, in particular with a gene insertion into HSCs in vivo. This is a glimpse of our pipeline, and what we have is really two franchise areas that we're pursuing. The first is in genetic diseases, and the idea is essentially to take any indication that can be treated with a hemopoietic stem cell transplant, and then more recently there have been a number of ex vivo approaches to some of these conditions, like sickle cell, but to take that and turn it into an in vivo hemopoietic stem cell engineering to treat the various different diseases. Our entry point into that franchise is chronic granulomatous disease, the X-linked form where the CYBB gene is defective. So we will insert a corrected CYBB gene into the HSCs, and then that will express only in the neutrophils where this disease is an issue. We expect, as I mentioned, to file this in the first half of the year and actually have clinical readouts in the second half. And so that's very exciting. We're also pursuing sickle cell disease, and that's a very large opportunity in this particular genetic disease franchise. And then quite interestingly, in an area where which is not treatable necessarily by HSC transplant, but we're going after oncology, but we're doing it in a way that no other platform can. So this is CAR therapy, but rather than just CAR in a single lineage cell type, like a T cell, like CAR T, but you also see CAR-M and CAR-NK as different approaches, some mostly ex vivo, some are approaching it in vivo with lentiviral vectors. we're actually going after multi-lineage cells. So we're CAR T, NK, and M, all three, to crack the solid tumor problem for the CAR approach, which no one has been able to do. And we're doing it in a durable manner because the HSCs are self-renewing and will continue to populate the myeloid cells, the NK cells, the T cells with fresh CAR-enabled cells, which has been a real problem for others where exhaustion has limited the efficacy. We can also approach the hematological malignancies with a CAR T and NK approach, where the M cells are not, the myeloid cells are not as important, and there we're seeking partnerships and active discussions. The advantages of the approach, which I've already alluded to, are essentially characterized here. The first one is very important. It is our high-capacity delivery, up to 35 kilobases, which is far more than others have been able to do. To be able to do multiple gene insertions and gene edits in a single product, for example, again, multi-lineage cell-specific expression. So that's a cassette for each cell type with its own promoter to make sure it turns on in the right cell. We can titrate after we've dosed without the need to redose because we have a cassette that allows us to do that, which is very important because titration after you've dosed, or essentially redosing, is a problem for all approaches due to the immunogenic nature of really everything, including lipid nanoparticles, and making it extremely difficult to redose. We can also add, for example, a kill switch, and we do have that if we want to undo the things that we've done. This is just overlaying those various advantages on different approaches to engineering, either in vivo or ex vivo, the hemopoietic stem cells or their specific lineage cells, and of course on the right, the ex vivo approaches. So I won't go through this in the interest of time, but we have significant advantages. Most importantly, we're actually doing something that these approaches cannot do. Nobody except Ensoma can do gene insertion in vivo into HSCs selectively. That is really what we do, nobody else can do. The virus-like particle that is at the heart of what we, of the program and the selectivity is characterized here. It is a chimeric particle with a capsid that comes from adeno5 and the fiber knobs that come from ad35, which have been modified further, so that's the plus. And this is a CD46 selective fiber particle, which allows it to be very preferential for the long-term HSCs in circulation. And so we're able to have our selective approach through that process. The viral genes have been completely removed from this, so it is a gutless adenovirus, otherwise known as a helper-dependent, because it needs a helper virus in order to replicate, and that's part of the manufacturing process. But it's very large capacity and very safe because all of these viral DNA that have been known in the past to cause issues have been removed. This gives a sense of how we improve the ex vivo processes that are otherwise used in this arena, either as a transplant approach or as ex vivo therapy. And it's a much, much better patient journey that is also far, far less dangerous, risky, expensive. The key things are the manufacturing process. We have an off-the-shelf, one-size-fits-all product rather than having to do a patient-by-patient manufacturing. And we do not need to ablate the hemopoietic stem cells in the bone marrow in order to install either the ex vivo therapy or the transplant. And the ablation process is very difficult, very dangerous, and that's something that we don't need to do. So it's a greatly improved, much simplified process, and that's one of the key advantages to our approach. The manufacturing is also, as I mentioned, it's an off-the-shelf approach, and this is another key advantage of how we approach this arena. Not only is it not patient-by-patient manufacturing, But the actual process itself is extremely straightforward, low cost, highly scalable, suspension cell based, very, very similar to antibody manufacturing and much less similar to AAV or other types of vector manufacturing that you may be familiar with. And so we are successfully scaled up to 200 liter. We're headed up to 2,000, and we think when we get to certain commercial scale for some of the larger indications, we can go to 20,000 liters. This can also be done essentially anywhere in the world. It doesn't have to be done in a highly specialized environment, at least with the technology that's currently available. So this is a key advantage to bring down the cost and to simplify the process for patients. So I can describe a little bit, starting actually with the immuno-oncology program, which is not the program that's headed into the clinic in the near future, but it's one that's very exciting because this is something that, again, nobody else can do, and is intended to crack the solid tumor CAR approach, which has so far been something that is not able to be addressed successfully. Our approach is this triple lineage cell approach, so CAR Ms, Ts, and NKs all together in a long, durable manner. So, and the CAR, as many of you are probably familiar with what the role of these different immune cells are, the CAR Ms would target the tumor, infiltrate the tumor microenvironment, and inflame the tumor microenvironment, making it susceptible to T cells and other cell attacks. And that's something that needs to be done in a durable manner because those typically ex vivo approaches have shown some very initial success, but then failed due to the fact that they wore off quickly. So you need a steady supply. And then you follow that with the CAR T and the CAR NK to then infiltrate the tumor and kill it. And so we're very excited about this approach. We're starting with HER2 as the target. The HER2, we have a number of reasons why we've selected that, but it's very de-risked biology. Some have said, well, this is a crowded field, but we are comfortable going after it because for us, it is not a crowded field. We are not an ADC. We're approaching this in a way that is extremely unique. So it's the same target as some others, but in a completely different mechanism, and usable where those are no longer effective. And so you can sort of see the construct on the right, which involves HER2 specific for the different cell type cassettes, along with this titration cassette that I mentioned that allows us to titrate up without the need to redose, as well as the kill switch that I referred to earlier. All of this is being built into this construct. This is some initial preclinical data that we've shared, just showing that our ability to control tumors in animal models using our approach. And so in each instance where we've dosed essentially with high levels of CAR therapy, but even with very, very low levels of CAR therapy, you get tumor control. And in specifically cold tumor models where the tumor is already initially established, we also show excellent tumor control. So we're very excited that this has tremendous potential in the arena and will be able to address the solid tumor challenge. Secondly, in the genetic diseases franchise, as I mentioned, we're going into the clinic this year with X-linked CGD approach. For those of you not familiar with chronic granulomatous disease, it is a rare disease where it creates dysfunctional NADPH in neutrophils. There are several genes that can contribute to this construct, and so there are several genes that can be defective. The most common one is the X-linked, where the CYBB gene is defective, and that's what we're pursuing in this particular indication. But we can pursue all of the forms of CGD, and we intend to do that as an umbrella approach to add the other forms. Patients who have this disease have very dysfunctional neutrophils and are highly susceptible to infections as well as inflammation. And it is often treated with HSC transplant, as I had mentioned previously, but there are challenges with that approach for many patients. Our approach is shown on the right, and it's very straightforward. We provide the corrected CYBB gene. with neutrophil-specific expression and the titration cassette that I had mentioned before. And just to jump to some of our early data in mice, this has been shown to be very effective on the left. The purple is our approach, which really has corrected for many of these mice that have the defective gene and made them quite similar to wild type. And this is very encouraging for us, and we're excited to bring this forward. On the right-hand side is actually a publication describing an ex vivo approach. This ex vivo approach is using the same construct that we're using, but they have to extract the cells and then ablate the system and insert the corrected cells back in. And this, they showed that this was effective. In fact, anything above about a 10% correction of CYBB produced very meaningful improvements in infections. So we're essentially taking this approach and moving it in vivo. This was on the right was a study conducted by Orchard, and so that's been published. And then thirdly, also in the genetic diseases franchise, I mentioned sickle cell disease. There are a number of companies pursuing sickle cell disease. There are approved ex vivo therapies on the market. Some patients do also, or instead of ex vivo, they've received hemopoietic stem cell transplants. So we know this is a condition that we can address and we can improve. However, the ex vivo approaches are having a difficult time getting established in the market. That's largely due to the incredibly burdensome nature of the procedures, the ablation. They're risky. Some patients don't survive that process, and that's in need of great improvement. Our approach on the right is, again, using our virus-like particle, but we're taking a base editing approach. And so we're reactivating fetal hemoglobin, which has been shown in the ex vivo approved approaches to work. So it's a very similar technique. We actually have a base editor that does not nick or cut the DNA, which we acquired when we acquired a company called 12 Bio. And so that's also exciting because I think that the dangers of potentially nicking and cutting DNA, which are quite common in most of the approaches, have not yet been fully appreciated. So we avoid the possibility of the hazards that will cause. And we also titrate up to effect without the need to redose. And that's what we're introducing through this process. This is just some very early data showing that we are able to, at a very high degree, successfully edit the appropriate genes and be able to achieve the level of HbF induction that's necessary to be therapeutically relevant. So we're also very excited about this area. Sickle cell is a huge problem globally, and our technique makes it much easier to treat patients, much easier to address the problems, and to be applied in a reasonable way that doesn't require extensive hospitalization for many patients throughout the world. So with that, that's concluding. If there are any questions, I think I'm out of time. I'm over time. Apologies to the next presenter. Thank you.